But we start off by talking about the fact that the capacity for the human body to digest and absorb the amino acidemia that comes from ingesting large amounts of protein actually far exceeds, as we'll come to see, the capacity for the skeletal muscle to utilize all of those amino acids to build muscle, to increase protein synthesis. So it goes on to say that following the digestion, the pancreatic proteases and enterocyte proteases break down the proteins into peptides and amino acids that then get absorbed by the enterocytes and go into circulation. So let's continue. So that's the first point. You should know that if you eat 40 grams of protein or 20 grams of protein in a meal, all of that protein is not directed to the skeletal muscle, as we'll come to see. The end products include peptides, as I just mentioned, fragments, and free amino acids that are absorbed almost exclusively in the small intestine. The gut is a highly metabolically active organ and extracts 40 to 50% of the available amino acids from the ingested protein meal, primarily for the purpose of energy production and for local protein synthesis. The remainder 50% of amino acids is released into the hepatic portal vein prior to being taken up by the liver. Like the gut, the significant proportion of amino acids are instead used for protein synthesis of hepatic and liver-derived blood proteins. So we get even more proteins and not proteins, but peptides and amino acids that are used by the liver and not actually making it to the skeletal muscle. The amino acids that have been sequestrated by the splenic tissue, and the liver are first passed clear, as I mentioned before, and thus are not available for peripheral metabolism. That means it never gets actually to the skeletal muscle. It is interesting to note that the branched-chain amino acids, the BCAAs, which are implicated in skeletal muscle anabolism, are catabolized to a relative minor extent by the liver due to a low content of the branched-chain aminotransferase enzymes in human hepatocytes. So this means that all of the amino acids that get sequestrated by the liver, there's a small percentage of it that are actually BCAAs, which, if you know, leucine is one or the most important BCAA, and it is actually the amino acid that sparks protein synthesis. And it's actually a very good thing that the liver does not sequestrate, I hope I'm saying that right, the BCAAs, primarily leucine. It's almost like the human body has actually evolved to leave the BCAAs alone because they are so important for skeletal muscle and protein synthesis. Although skeletal muscle is a large depot for the retention of amino acids, not all amino acids released into the plasma are destined to become incorporated into new skeletal muscle tissue. So even though the amino acids that actually make it to the bloodstream, not all of them, or to be fair, most of them are not actually to be incorporated into skeletal muscle and hypertrophy or protein synthesis. As we'll come to see, in a recent study employing an intrinsically labeled tracer approach, Groen and colleagues demonstrated that only 2.2 grams or 11% of the amino acids provided to young men in a 20 grams bolus of casein protein were used for de novo protein synthesis, despite 55% availability in the peripheral circulation following splenic extraction. So what does this actually mean? It means that of all of the protein that was actually ingested, the 20 grams of casein protein, only 2.2% of that whole protein actually made it to the skeletal muscle. So that's 11% of the whole protein that gets used up by the skeletal muscle. So that's a tiny fraction of the actual amino acids that make it to be incorporated in the skeletal muscle to be used as protein synthesis and form new proteins, myofibril and sarcoplasmic proteins, and so on and so forth. So as you can see by this first chapter, the idea that when you eat a lot of protein, most of that protein actually makes it to the skeletal muscle and it's being used as protein synthesis to enlarge your muscle fibers is actually very, very wrong. Okay, so 50% of all of that protein actually gets eaten up by the liver, and then the remaining amino acids that actually make it into the bloodstream, some are utilized to make blood proteins, like albumin, for example, and a lot of the other amino acids actually are used up by other tissues. We don't have only muscle mass. We have a lot of other organs and tissue that actually needs proteins, amino acids, like let's say to build up tendons for collagen production or for other soft tissues. And we get left with 11% of all the amino acids provided being actually intaken by the skeletal muscle. So that's a very tiny percentage of actually amino acids that get incorporated into the skeletal muscle. So now that we have actually learned that the liver is kind of a jerk, gets all of the amino acids first, and then we have the urea and the albumin and all of the blood proteins and all of the other tissues and organs that get their fair share. And by the time that the amino acids actually get taken up by the skeletal muscle, we only have 2.2 grams. What a sad day. What a sad day to be a muscle fiber. Moving on. Regardless of the protein source, the feeding induced rise in plasma amino acid concentrations drive uptake across the muscle membrane. So, despite what is actually your source of protein, you may be eating beef or whey or casein, the feeding induced rise in plasma amino acid concentrations actually get uptaken across the muscle membrane, and that is a good thing, right? Some evidence suggests that protein ingestion induces gene expression of several amino acid transport proteins that may increase the influx of amino acids into skeletal muscle. So the skeletal muscle actually senses the rise, the correct term for this is the exogenous rise in amino acids that are in circulation, and it elevates the genes of the amino acid transports that actually pull the amino acids into the muscle fiber, which is a very good thing. And again, biology wins again with the evolution. Following substantial hyperaminoacidemia, there is a 30-minute delay in the stimulation of muscle protein synthesis before it peaks at 2 hours. So we get a half an hour delay, and then it peaks after 2 hours. Typically, this can vary based on the type of protein that you ingest, right? Because we have fast-acting proteins and slow, so that can vary, but it's a good place to start.